So here comes physiology of the digestive tract or digestive system. So we're going to look here. We're going to have our phases of gastric secretion. This side is stimulatory events. This side is inhibitory events. Up here, we're going to have stuff that happens from the thought process. Here, we're going to have stuff happening from within the stomach. And here, we're going to have the effects from the intestines. So we call this the cephalic phase, as in cephalad, as in head word. We call this the gastric phase, which means stomach. Then this is the intestinal phase, like intestines. So first off, stimulatory events, pre-food. I like apple pie. My mother-in-law makes amazing apple pie, and I sit and I think about her apple pie quite often. I'm thinking about it right now, and I'm getting kind of hungry. It's making my mouth water, and if I keep thinking about it for a while, my stomach's going to start to growl. So what's happening? I'm thinking of food, okay? As I think of food, I have a conditioned reflex. I've had her apple pie many times. It's amazing. And I have a hypothalamus and medulla connection. So I have an emotional connect to it. It is good, and I associate it with good times. What does that do? My vagus nerves sends information to my stomach, and it says, get ready for some apple pie. Start making some acids and juices, and start churning up and be ready for it. If I were to smell the pie, that would be an even stronger stimulation. If I ate the pie, then it's going to stimulate the stomach even more. So now that I've eaten the pie, let's say it is traveling down my esophagus, passes through my cardiac sphincter, and now it's in my stomach. When the pie comes into my stomach, the stomach will distend, and when it distends or stretches out, there's going to be these vagal reflexes, and that is going to tell the stomach start secreting more stuff. And there's also going to be local reflexes, and that's going to tell the stomach start secreting more stuff. When the food gets there, alright, the chemicals from within the food, they're going to change the pH. They're going to make the pH rise. They're going to make the pH become more basic. So what does that do? That tells the G cells in the stomach to release gastrin into the blood, which will really get the stomach going and really get the stomach releasing its acids and its enzymes. After the food's been in my stomach a couple hours, it starts to break down, it starts to get released into my small intestines. Just a little bit at a time. You can't release it all into the small intestines or else it'll damage it. So as we release our little bits of chyme from the stomach to the small intestines, that presence of low pH and partially digested foods and fats will cause the small intestines to release gastrin into the blood and get some more stuff going on, alright? Now, with our inhibitory stuff, as we get distension of the duodenum from the presence of fatty, acidic, hypertonic chyme, what that's going to do is that's going to have local reflexes, and it's going to have vagal nuclei in our pyloric sphincter, and those are going to tell the stomach, hey, you're starting to release food into the small intestines. I don't think any more food is coming to you. There's no need for you to keep on producing stomach acids. So let's stop with the release of stomach acids. That's the gist of what the little reflex from the small intestines to the stomach will be. Next, with the distension of the small intestines and the presence of all those stuff, we're going to have some release of hormones, secretin and cholecystokinin. Those inhibit the gastric secretion, and those are going to help with the secretion of bile and stuff from our pancreas. Okay? If the stomach gets too acidic, that's going to tell your body, your G cells, to stop releasing gastrin. If you get emotionally upset, that is going to change your hunger level too. Alright? And that's going to be right up here. So loss of appetite or depression. So right now I'm thinking about apple pie, and it's hungry time, and yay, I'm getting a little hungry. But if somebody told me my fish died or something, it might upset me, and I would get depressed and I would get upset, and that would make my appetite become suppressed. Okay, so within the stomach, there's going to be 4 digestive activities. There's going to be receptive relaxation, gastric accommodation, gastric contractile activity, and regulation of gastric emptying. So those are going to be straight for your study guide. That's why I stuck those on there for you. So first off, when food comes into the stomach, it is going to need to relax to receive the food stuff coming into the stomach. Once the food comes into the stomach, it is going to need to accommodate it. So how does it accommodate it? It has gastric secretions. So that's where our gastric accommodation comes into play. And the next one is gastric contractile activity. So what's happening there? The stomach is contracting and it is mushing around the food and mixing it with the enzymes and acids in the stomach to break down those proteins and get them ready to go be absorbed in the small intestines. And then the last part, regulation of gastric emptying. Like I said, the stomach doesn't just push all of the chyme from the stomach out into the small intestines all in one big pop. It just does a little bit at a time. And the reason for that is so it doesn't damage the small intestines, and also so you're going to have an easier time absorbing all of the goods, fats, and nutrients and sugars that you're getting from the stomach. Okay, so let's follow a drop of food from the mouth through the digestive tract. So I ingest my food. I begin mechanical digestion by mastication with my teeth, and some chemical digestion can occur in the mouth from salivary amylase. I swallow my food, and it will begin its propulsion through the esophagus. Travels down the esophagus through the cardiac sphincter, or the lower gastroesophageal sphincter, and it will enter the stomach. Gastric receptive relaxation occurs to let the food in. Gastric accommodation occurs to keep the pH of the stomach stable. Gastric contractile activity begins to beat up and pummel that food, and then we're going to get our output of the food from the stomach to the small intestines. So let's go through our parts of our stomach. There's the fundus. There's the cardiac region here. Then we go into the body of the stomach, and the pyloric region, which is the funnel of the stomach. Then we get into our small intestines. Alright, so we pass through the pyloric sphincter to go to our small intestines. First third of the small intestines is the duodenum. Most absorption of foods, of nutrients and fats and glucose will occur in the duodenum. The reason for it is this ampulla of Vater that we are going to speak of. What happens in that first third of the small intestines is you release the bile and all your enzymes to really break down the food and get it ready to be absorbed. Alright, so we go duodenum is the first third, second third is the jejunum, last part is the ileum. And then we go into our colon. There's the cecum, the ascending colon, transverse colon, descending colon, sigmoid colon, and the rectum. So here's this hepatopancreatic ampulla of Vater, or hepatopancreatic, whatever. It goes by a bunch of names. We got an ampulla, we have a sphincter, we have a papilla. When we see this ampulla, the ampulla would be a bump. So here they're showing the smooth muscle sphincter, and the papilla is the hole. Over this there will be mucosal tissue. That's going to be called the ampulla. Alright, so the papilla is the hole, the sphincter is the smooth muscle, and the ampulla is the bump. This is called the major duodenal papilla. Okay, or it can be called the ampulla of Vater, or it could be called the sphincter of Oddi. Here's what's going on here. Up in this region right here where I'm kind of circling with the mouse, this is where the liver is. The liver is going to be drained by the right and left hepatic ducts, and those guys come together to form my common hepatic duct. Sorry, my common bile duct. Common bile duct meets up with the cystic duct to form my bile duct. Alright. Then we have my bile duct sphincter. So if right now I haven't eaten in a little bit actually, and my liver is possibly producing bile. If it's producing bile, it comes over here. There's no fats in my small intestines to be absorbed, so my bile sphincter is contracted. So what happens is the bile is going to have to regurgitate, and it'll get pushed back up into the gallbladder. That bile stays in the gallbladder and it gets concentrated until I eat a fatty meal, and then this sphincter of the bile duct can relax and my gallbladder can contract and give me a nice burst of bile to help break down fats. Alright, as you see here, the bile duct and the main pancreatic duct meet up to form this hepatopancreatic duct, and then we get the ampulla and the sphincter and the papilla and all that stuff, but these guys are going to meet up here. So here's my pancreas. We'll have our main pancreatic duct, and it has an offshoot of an accessory pancreatic duct. That's there as like a backup mechanism if something was to occur to the main pancreatic duct, or it's there just if there's a little extra contents to release. So here's our microscopic features of the small intestines. We're going to have plica circalis. We'll have villi, which will be finger-like projections coming off of these plica circalis. Here's our villi. And we'll have microvilli. Plica circalis will be there to slow down the food and to create turbulence. So to slow down the chyme traveling through the small intestines and to create turbulence. Why? We want to absorb stuff. In order to absorb stuff, it needs to be traveling at a very slow pace. Next, it's going to create turbulence, so the chyme is being mixed up, so all the stuff from just the nutrients aren't getting absorbed from outside of the chyme. It's getting middle outside, and it's just continuing the mixing process. Next ones are villi. The villi, along with the microvilli, are going to be around to increase surface area of the small intestines. The larger the surface area, the more nutrients, fats, and sugars we can absorb. So the way I like to think of this is if you had like a hill, if we just had a flat land, that could be the least amount of surface area possible. Okay, like 1 square mile by 1 square mile. All of a sudden, if you put some hills in there, those could count like our plica circalis. They're nice big bumps. On top of those hills, let's put some trees. So the trees would represent the villi. They're increasing the surface area of this 1 square mile by 1 square mile even more. And then on top of those trees, let's put leaves. So the leaves would represent our microvilli. There's tons and tons of surface area from the leaves of a tree versus just 1 flat square mile. Okay, next part's your liver. So here's some liver anatomy. Here's your right lobe of the liver. The bare area falls in the right lobe of the liver. You have your falciform ligament, which separate the right and left lobe, and once it comes off of the liver, it is called the round ligament, or the ligament of Terry's. And then you get your left lobe of the liver. Just inferior to the liver and lateral to the round ligament is going to be the gallbladder. We're looking at an inferior view of the liver here. Alright? So here's our gallbladder, here's the sulcus for the inferior vena cava. Okay, here's the quadrate lobe of the liver, here's the caudate lobe of the liver. Within the lobes of the liver, there are lobules. So lobe, certain area of the liver, lobule are going to be our functional units. They will be hexagonal in shape, and at the corner of each, at each of the 6 corners of each hexagon, there will be a portal triad, and there will be a central vein. Inside of these, we will have our hepatocytes. The function of those guys are going to be all these little cells here are the hepatocytes, the brown ones. The functions of the hepatocytes are going to be to produce bile, process nutrients, store fat-soluble vitamins, and to detoxify any junk. So here's the gist of what's happening. We're going to have our interlobular veins. All right, those guys will let these lobules talk to one another. And what will happen is we'll have this central vein that will collect blood traveling towards the midline. So as I said, at the corner, each corner of the hexagon, we're going to have a triad. In that triad, we're going to have a portal artery, portal vein, and a bile duct or bile canal. The artery and vein are going to be bringing in goods to our hepatocytes and unprocessed goods to the hepatocytes. So that stuff will travel from the triad into the central vein. Here we're going to have a canaliculus to collect the bile being produced by the hepatocytes, and it will flow into our bile duct here. Alright. Also floating around we have these Kupffer cells, which will be macrophage found specifically in the liver. Why? Because we're going to have leaky capillaries and we're going to be able to let large stuff through, and there's going to be a high risk of microorganisms passing into there. Okay, next one, we already talked about the gallbladder, right? So we have our liver, which would be sitting up here. It produces bile. It gets drained by the right and left hepatic ducts. They turn into the common hepatic duct. Common hepatic duct will combine with the cystic duct to form the bile duct. And if that bile duct sphincter is contracted, bile regurgitates into the gallbladder, and the gallbladder will concentrate the bile. Alright. Next one is our pancreas. So our pancreas has endocrine and exocrine functions. Its endocrine functions would be its insulin and glucagon. Its exocrine functions will be its enzymes. So those will be proteases to break down proteins, amylases to break down carbohydrates, lipases to break down sugars, and nucleases to break down nucleic acids. So let's talk about a little bit about hormones here. We're going to have two of them. We're going to have cholecystokinin and we're going to have secretin. Okay? Cholecystokinin is going to be released in the presence of fatty foods. Alright? Secretin is going to be released when we have more acidic type foods. Alright? So, 1. Acidic chyme entering the duodenum causes enteroendocrine cells of the duodenum to release secretin. So acidic chyme gets us to release secretin. Fatty or protein-rich foods will get us to release cholecystokinin. Either or, secretin or cholecystokinin will enter the blood, and when they come to the pancreas, cholecystokinin will induce the pancreas to release enzyme-rich pancreatic juice to break down fats, proteins, carbohydrates, all that stuff, and secretin will tell the pancreas to release bicarbonate. Why? Bicarbonate's a buffer, and it'll help decrease the pH of the chyme to make the small intestines less acidic. Okay, here's our large intestines. So the ileum of the small intestines meets up with the cecum of the large intestines. The connector between them is the ileocecal valve. Alright, so here's the cecum. This is the appendix, or vermiform appendix. Cecum guides into the ascending colon. We get a right colic flexure, transverse colon, left colic flexure, descending colon, sigmoid colon, and then rectum and toilet. These pouches are called haustra. These little fat droplets coming off are called epiploic appendages, and this line of muscle traveling across here is called the tinea coli. With the anus, there will be an external anal sphincter which is circular, and there will be an internal anal sphincter which goes up and down. The external anal sphincter is under conscious control. The internal anal sphincter is under subconscious control. So when you get the urge to defecate, you're having contraction of the colon and the rectum, and the internal anal sphincter is relaxing. If you do not defecate at that moment and you decide to hold it, your external anal sphincter is going to be contracted because you have conscious control over that. Okay, so what's our large intestines do? It's going to help absorb water. It's going to help absorb vitamins B and K, and those will be produced by some of our bacteria. Okay, so we'll have our external anal sphincter, which will be circular fibers, and we'll have our internal anal sphincter, which will be longitudinal fibers. And then here we have our hemorrhoidal veins. So if you guys have heard of hemorrhoids, What a hemorrhoid is imagine this. This is on your rear. So if you are sitting, you're sitting for a long time, there's pressure on these blood vessels, all right? Or if you're bearing down, or you have a heavy weight like you have a baby pushing against your pelvic floor, there's pressure on these blood vessels. Well, blood vessels are low pressure with blood, right? They have very low blood pressure. So if we're squeezing these, we're pinching these, blood can't get out of them. So what happens? Blood starts to pool in them, and veins do what they do best. They act as a blood reservoir. They start to stretch out and stretch out and stretch out. As they stretch out over time, all of a sudden they can no longer vasoconstrict, and they lose their shape. So as they lose their shape, they can start to bulge, and they can literally be coming out of the rectum, right, or out of the anus. This is your rectal valve. There's going to be a series of rectal valves, and the function of the rectal valves are to allow the movement of air or gases instead of the movement of fecal matter. So you can pass gas instead of defecating. Alright? Your lesser omentum comes off of the lesser curvature of the stomach. Your greater omentum is going to come off of the greater curvature of the stomach. Okay, from these, what do I want you to know? So here's our carbohydrates. I want you to know where stuff is occurring and what is released in each area. So for example, with carbohydrates, the mouth releases salivary amylase. The small intestines releases pancreatic amylase. Once we get into the brush borders of the small intestines, we get specific enzymes for each type of sugar. Alright, so we'll get our dextrinases, glucoamylase, lactase, and maltase, and sucrase. With proteins, digestion does not occur until you get into the stomach, and that will be activated pepsin from the presence of HCl. So HCl activates pepsinogen into pepsin. In the small intestines, we're going to release our trypsin, chymotrypsin, and carboxypeptidase. And then we move further into our brush border enzymes with our aminopeptidase, carboxypeptidase, and dipeptidase. Alright, next step, we have our fat digestion. That's going to occur in the small intestines, and bile from the liver will be released. Alright, bile is stored once again in the gallbladder. And then we'll have pancreatic lipases to really break them down. Next one, we're going to have nucleic acids. Those guys are going to be broken down by ribonuclease and deoxyribonuclease. Ribonuclease breaks down RNA. Deoxyribonuclease breaks down DNA. Right? And then we get our nucleosidases and phosphatases. That's it.